Good afternoon and welcome to the CERIAS Computer Security Seminar from Purdue University. Our speaker today is Emiliano Di Cristofaro. Did I get that close enough?
Perfect.
I'm sorry.
No,
He's from PARC, the Xerox company, the famous research center there. And his topic today is whole genome sequencing, innovation dream or privacy nightmare. Emiliano?
Thank you very much. Thank you so much for hosting me and for the introduction. I'm really honored to join the list of renowned CERIAS seminar speakers. So my talk today is going to be about whole genome sequencing and privacy. So before going into the Before going into the details, I'd like to give a brief introduction of whole genome sequencing technologies, starting with a brief crash course on genomics. So I'm confident that most of you are familiar with this topic, so I'm going to cover them quickly for those who are not. So a genome is really a container of the entire hereditary information that is needed to build and maintain a living example of an organism. It's encoded in DNA or actually RNA for certain kind of viruses. And I'm pretty sure you're familiar with DNA, this double-stranded polymer of nucleotides, these letters A, C, G, and T. So in humans, in human genomes, there are 3.2 billion of these nucleotides organized in 23 chromosome pairs. We inherit one chromosome from our mother, one chromosome from our father. So whole genome sequencing is really the science and the technology aiming to determine the complete DNA sequence in an organism genome. Right. And so in the last few years, a little bit more than few years, whole genome sequencing, or I'm going to use the acronym WGS, has attracted really a lot of attention and a lot of investment. So just let me give some brief history. DNA sequencing really started in the 70s, but it was only in 1990 that the prominent Human Genome Project started. It was a big collaboration, I think was started by the US Department of Energy and the National Institute of Health with collaborations with different countries as well. And it took 13 years to actually complete the sequencing of the first human genome. And after that, everything started to go pretty fast. So in 2010, a race to lower the price to $1,000 for sequencing was actually completed. So it's very interesting to see how prices for WGS technologies really dropped very, very fast. So the Human Genome Project was an investment of $3 billion. But around 2010, actually, after the advent of high throughput sequencing technologies, we really went down to affordable prices, so lower than $5,000. So recent announcements, certain companies claim that they can do that with less than $1,000, even $100 perhaps. But whether or not we will keep these promises, it is interesting to notice that really WGS technologies are about to be ready for the masses. So it's not really far-fetched to imagine a near future, maybe 5-10 years from now, when where most of individuals will have access to their sequenced genome. So if you think about it, prices could be lower than getting an MRI, and then you actually have to sequence your genome just once in your lifetime. So in this talk, I'll try to cover both the good news and then the bad news stemming from this revolution. The good news is mostly related to the effects that WGS technologies will have on medicine and healthcare. So we will talk about this dream of P4 medicine, where these 4 Ps stand for predictive, preventive, participatory, and personalized. We will talk about genome-wide association studies, but we will also cover some serious privacy, security, and ethical concerns that stem from all of this. So we start with personalized medicine, which is a very generic term that refers to the fact that we could tailor diagnosis and treatment of many diseases to the patient's genome. This has two components. One is that having patient's genome fully sequenced will actually help doctors to better understand the disease that the patients suffer from, and also determine a much more effective medication. So this is something that happens already today. There is a number of tests that are done, especially for certain kinds of cancers, leukemia, but there is much more than that. So if you think about it, if we have a genome that is already sequenced, so we have it, let's say on a computer or something like that, running this kind of test will be much more cost effective, much more efficient, and in certain cases even more accurate than how tests are done today in lab, where we have to collect a sample, send it over, and use some very expensive, sometimes very expensive lab equipment. There is more to that. Certain kinds of tests can be classified as preventive medicine. So we can look at genomic information to understand the predisposition to certain kind of diseases. We can test for first early symptoms of Huntington disease, diabetes, and so on. There is more and more interest in newborn screening based on genetic information, and this is actually something that you probably have noticed already with certain commercial offerings that are becoming increasingly popular, like 23andMe, that offers you for, I think, a couple of $100 a genetic screening. So they will give you a report telling you about the predisposition to certain diseases and more. So from a computer science point of view, really we can look at genomic tests as an algorithm problem. So we can design computational tests, so we use the term in silico, to simulate or to perform tests that are typically done in lab, right? So as I mentioned before, in a much more efficient and cost-effective way. So just to give you some more concrete example, so paternity test is something that you can do today with, I think, $39. You can buy a kit at Walgreens and send some samples to a lab facility. But if we have genomes already sequenced, we can actually run some algorithms that either scan the entire genomes or really emulate tests that are done today in lab. And I'm actually going to talk about it in a few slides. We can go a step further to beyond paternity tests and look at ancestry and genealogy. So these kind of testings that are done to trace one's lineage. This is very helpful in certain medical applications, but again, there is an increasingly popular market for this kind of testing, and I'm pretty sure you have heard of Ancestry.com, where again, they look at your genome and they try to infer your ethnical heritage and try to find your ancestors, things like that. Again, more toward medical applications, other tests that are commonly done today are compatibility tests, where, for instance, two potential partners want or need to assess the possibility of conceiving a child with a recessive genetic disease. This happens where you have one chromosome that has a mutation, one chromosome that doesn't. If both partners have these mutations, there is a non-negligible probability that both chromosomes will be affected by the mutation, and in that case, certain kinds of diseases are really life-threatening. So you need to assess this. But actually, there are, again, companies that run some obscure compatibility tests where they try to assess the probability that you are a good couple. I don't really know how they work, but you can go on genepartner.com. They offer this service where they have an improved matchmaking algorithm based on genetic information. And as I mentioned before, there's also an increasing interest on genome-wide association studies, where we really transition from algorithms problem to machine learning, AI. The idea is pretty simple. Again, this is a very generic term, but the idea is if we have if there is an increasing availability of a large number of genomes, annotated with medical information about, for instance, diseases and response to treatment, we can try to find correlation between genetic features and diseases, right? So this is sort of analytics, just to use another buzzword. So now I've covered some of the good news that come with WGS, the revolution in WGS, but let's also look at the bad news and the privacy and the ethical concerns. So the first observation is really that the genome is the ultimate identifier. So it really uniquely identifies its owner. So, I mean, once it's leaked, you cannot revoke it, right? Like you would do with a stolen password or a stolen credit card. And this is particularly bad because genomic information contains very sensitive, the genomes contain very sensitive information about, for instance, I'm just giving you some examples, ethnic heritage, predisposition to disease, even certain mental conditions are correlated to certain genetic features. And actually, this is bound to get worse because we're discovering every year more information about genomes, more correlations about genes and certain kind of diseases and so on. So I actually read an article during the campaign time where some researchers have correlated certain genetic features to the way people vote, for instance. Okay. And this actually, these fears had been already explored before the revolution of WGS. And if you remember, there was a movie in the 90s called Gattaca, that really depicts the risk of genetic discrimination. So it's, of course, it's a Hollywood movie, it's a sci-fi, but it raises some very interesting questions. So I recommend to watch it if you haven't. More importantly, actually, I wanted to point out the fact that we could think of anonymizing genomes so that we remove the information about the owner. We remove the binding between the owner and the genomic information. Unfortunately, this doesn't really help. As you can imagine, genomes are very uniquely identifiers, and there's been a couple of results in this direction. For I think 1 or 2 months ago, there was an article in Science where researcher had looked at a database of available genomes. So, many people have donated their genomes to science, so to allow researchers to do certain studies. And they were able to de-anonymize these genomes by correlating genomic information that was available in some other so the external sources, so other information like the one in Ancestry.com and so on. So I think their accuracy was above 80%. And this was something that had already been pointed out in 2008. So just to clarify, my point of view, it's even worse than that. So, and the fact that genomic information has some peculiar characteristics. So if you leak your genome, you're actually leaking the genome of your close relative as well. And this comes from the fact that 99.9% of genomes of closely related humans is actually identical. And this actually was the basis of this attack published in Science. So there was even some lawsuits because of that. As I mentioned before, many people have donated their genome to science for research purposes, and some of their relatives didn't really like that and sued the relative. And finally, the last point is that really the sensitivity of human genome, because of this hereditary characteristic, it's almost perpetual, right? So if my genome gets leaked 60 years from now, I might not care that much. I'm probably dead, but my relatives, my children might, as I mentioned before. So this becomes also a question in terms of the effectiveness of traditional security mechanisms like encryption. So usually we tend to trust encryption mechanism for 30, 50 years, but this might not be enough. So this makes very hard to realize genome-wide association studies. As I mentioned before, they really rely on the availability of a large numbers large number of genomes. So if we cannot create these databases where people donate their genome, then it's going to be very hard to implement that. So from the legislation and policy point of view, there's been some initiatives. I just thought it was worth mentioning. Genomes, they're considered health information, so they're protected by HIPAA, the HIPAA legislation, like any other kind of health information. But there's also one law in the US, it's called GINA, that really tries to talk about non-discrimination based on genetic information. Certain US government agencies have funded research to investigate ethical, legal, and social implication. Some non-profit organization, non-government organization have issued certain recommendations. And I really encourage you to read a recent report by the Presidential Commission for Study on Bioethical Issues. Is written in layman term and really motivates some of the ethical concerns and calls for stricter regulation on whole genome sequencing. Again, from the point of view of computer science researchers, there is not only privacy challenges that makes WGS a nice field to work on. So for instance, one is how to efficiently design computational genomic techniques that deal with whole genome. So as I mentioned before, in a human genome there are 3.2 billion letters. So if you try to design algorithms that work on such a big input, you really need to care about efficiency. Okay, no, not very surprising. But we should also start posing questions that deal with how to design systems supporting genomic operations. So we have to answer a very basic question. So we have centers that can sequence whole genomes for a reasonable price, but where do we store it? How do we protect it? How do we give control to the owner of the genome itself? So there's also, I would say, more ethnographic based questions, HCI, really the challenges that we have to face. So in our work, we really try to answer some of these questions, try to take a first step, and we wonder whether or not cryptography can help to protect privacy in genomic tests. So what we want to try to do is to design privacy-preserving tests on whole human genomes. Really, our goal is to protect privacy, and let me clarify what I mean. So we would like individuals to really remain in control of their genome. So have it under control, but still they could allow doctors or clinicians or some labs to run genetic tests. But the privacy goal is to do so in such a way that only the required minimum amount of information is disclosed during the process. So that means if I need to test for certain kinds of mutation, remember the examples for personalized medicine, where we need to test for the presence of certain mutations before receiving certain drugs for cancer, I don't want to necessarily reveal my entire genome to the doctor or to the testing facility. I just want them to know whether or not I have this mutation. And our approach is to rely on some secure information sharing, cryptographic protocols. I will give a brief overview of our approach to show our intuition. The other goal is really efficiency. So in cryptography, there is a number of techniques that they're very powerful. They can solve many problems. So in theory, we can show that this kind of tests can be done with privacy, but we need to have reasonable efficiency. And this is particularly hard because of the size of the input. So we're dealing, again, with 3.2 billion letters. So we need to find ways to reduce the size of the input to the cryptographic layer, and to try and leverage as much pre-computation as possible as well. So the first step was our paper published at CERIAS, where it was really the first work to focus on whole genomes. And in that paper, we show how we can provide privacy-preserving tests for some common applications like paternity test. We kind of use it as a measuring stick. Is probably the easiest test and the one that most people can relate to. And then we look at personalized medicine and genetic compatibility as well. And the intuition is to realize the privacy layer is to use cryptographic protocols that are known as private set operations. So in particular, private set intersection. So let me give you an overview of how these protocols work, and then I'll show you how you could implement genomic tests with privacy by relying on these protocols. So private set intersection is a very simple cryptographic primitive that involves two parties. Let's call them server and client. They have some private data sets in input. These data sets can be these items in the data sets can be anything: numbers, identifiers, strings, nucleotides. And they want to compute the intersection in such a way that one party receives the intersection in output and the other doesn't get anything at all. So that means that nothing is disclosed beyond the intended result. So if you had a trusted third party, it would be trivial to realize such a protocol. But the goal in these cryptographic protocols is to replace the trusted third party, but with a protocol, right? So you essentially realize the exact same functionality. So there's been some recent advances in private set intersection. Techniques to realize this kind of computation had been around for many years, but it was only in 2010 that we proposed the first protocol that has linear communication and computation complexity. Just to put that in perspective, prior work had quadratic complexity, so it was hard to scale to large sets. And we have a couple of constructions that you can look up if you're interested. Security in a honest but curious model, in malicious model, and in particular, we released an optimized implementation that uses pipelining and shows how to properly implement these protocols. On the other hand, there's been also some work leveraging garbled circuits. The garbled circuits are generic techniques that can realize any sort of functionality with privacy, so some functions that can be expressed as a Boolean circuit. There's also been some advances in this direction, so by tailoring circuits to PSI. However, there are still a few orders of magnitudes lower than the specialized constructions. Also, beyond the simple, besides the simple private set intersection construction that I showed you before, there are certain extensions that can become very useful in real-world applications. So we worked in this area as well. So extensions that you may think of is disclosing only the cardinality of the set intersection instead of the actual content, introducing a mechanism to certify inputs to be policy compliant in the protocol, or hiding the size of sets. So if you're interested, we can talk about it later as well. But now I really want to show you how to leverage these techniques to realize private and efficient testing on whole human genomes. We will start with paternity test. Again, it's probably not something that we envision as a product in the future, but it serves as a measuring stick. So the paternity test is probably the easiest genetic test that we can realize. So we want to see if we can do it in a privacy-preserving and efficient way. And then we move to more perhaps interesting applications that deal with medical scenarios. So one interesting observation is that paternity test today is done in lab. You collect a sample, you do certain operations on this DNA sample, and then you declare whether or not the test is positive, right? But now, if you were to implement this algorithm, so this test in computation, and you have the entire human genome of the alleged father and the alleged child, you could do something very, very accurate. You could just compare the genome of the the candidate with the alleged child, and say that the test is positive if the number of matching nucleotides is above a certain threshold. And this relates to the fact that 99.9% of the genome of a father and the child would be identical. Whereas if you take the genome of any two individuals that are not related by a father-to-child relationship, this percentage is much lower. It's 99.5%. And remember, this is 3 billion the size. So this 0.4% is actually a significant difference. So now, if we were to implement this protocol in such a way that privacy is protected, so that the alleged father and the alleged child do not have to disclose the entire genome to each other or to any other involved parties, like a test facility and so on, we could do that. This is a simple comparison. We just have to count how many nucleotides are matching. So we could do that with certain secure two-party computation protocols, including private set intersection cardinality, right? So this will give us a very accurate test, will be really resilient to error, but the performance are not promising because, as I mentioned again, the human genome contains 3 billion symbols. So we actually try to run this protocol on some genomes that we collected from publicly available research databases, and it took a few days. So we tried to improve on this, and we really realized, okay, I just said 99.5% of any two human genomes is identical. So why don't we compare only the remaining 0.5%, right? So if 0.4% of this 0.5% is identical, then the test is positive. Otherwise, it's not, right? And this, again, it could be done with privacy, and now it could be done even efficiently because we reduce the size of the input from by 2 orders of magnitude. And in our experiments, this would take about an hour, which is acceptable for a paternity test. But the problem is that we don't know where the differences in the human genome occur. So I said 99.5% of any two human genomes are identical, but I cannot predict where this 0.5% is going to occur. So now how we do it for real is we really emulate what's going on today in lab-based tests. So lab-based tests today, they're actually admissible in court, use a couple of techniques actually. One of them is based on RFLP, restriction fragment length polymorphism. It's a very simple test. So in lab, they take a sample of DNA, they cut it into fragments using some enzymes, and then 25 of these fragments are selected using some markers. It's a process called electrophoresis. Now the test is positive, so we say that the sample of alleged father is a positive against the sample of the alleged child if at least 24 out of these 25 fragments have the same length. And this is accurate enough to be admissible in court. So now, as you can see, it's not very hard to emulate this protocol, this test in computation, right? The cutting part can be done by simple pattern matching algorithm, and the selection as well. So the markers you can simulate it by using a pattern matching. Now I want to run this test in a privacy preserving way. That means that I just want to check how many of these fragments have the same length without revealing any content, without revealing the length of the fragments if they're not matching. So again, I can use techniques like private set intersection cardinality. I just count privately how many fragments have the same length. So a few remarks. When I presented this work, a few people have asked me, why do you compare fragment lengths and not the actual contents, right? So, intuitively, it would be more accurate to compare contents. But in reality, the way it works today in biology, in biological-based tests, is that RFLP yields very little false positives. So there's no harm in comparing the length, but actually we have an advantage because in case there's been some sequencing errors, so some letters were changed from A to C or something like that, we will be resilient to that. So we've done some experiments, and just to give you an idea of the efficiency of these protocols, so it will take about a minute on a desktop to, or actually I think it was my laptop, to run the preprocessing phase, so to cut these 3 billion letters into some smaller fragments and select some of them. But then the real-time computation, so involving 2 interactive parties, will only take about 10 milliseconds or so. I mean, the comparison is really, the input of the comparison is really small, just 25 fragment lengths. So we can actually implement this kind of tests even on a mobile device if we want. And the interesting thing is that actually we ran some studies. So we partnered with bioinformatic experts, and they told us that if you select 50 fragments instead of 25, your accuracy will go from 99.9% to 99.999%. So you can just double the amount of computation that you do, but have 2 orders of magnitude more in accuracy. Then we look at personalized medicine. Again, as I told you before, it's an approach to, for instance, choose a treatment plan depending on the patient's genetic features, or to choose one drug or the other based, again, on some genetic fingerprints. And just to give you an example, before prescribing certain kinds of drug for leukemia and HIV, we need to test whether or not the patient has some kind of mutations. So in a way, we can say that we need to test a drug's genetic fingerprint against the patient's genome. So similarly to what I said before, the privacy goal in this kind of tests will be to let patients retain control of their genome, so they don't have to disclose it to the doctor or to the test facilities, but still let the doctor know whether or not this mutation is done. So this can be actually even more complicated than that, because certain pharmaceutical companies protect the intellectual property of these tests. So they don't want to disclose what these tests are, and there's been a recent lawsuit in this area that the company has won. And even to complicate it even more, yes, the company might be able to conceal the test, but still needs to have an approval to run certain kinds of tests. For instance, in the US, any medical test needs to be approved by the FDA, the Food and Drug Administration. So with all these constraints in mind, we find a way to reduce this kind of testing for personalized medicine with privacy, using another private set operation protocols, in particular authorized private set intersection. So the company that runs this test needs to certify that its inputs are compliant to a certain policy, and the certification comes from the FDA. So it's a pretty complex problem. I'm going to skip the details for the interest of time, but you can read more on the paper. So last 2 tests that we support are compatibility test and ancestry test. Compatibility test, we don't do the online dating algorithm, but we look at the partners that want to assess the risk of conceiving a child with a recessive disease. And this is actually an example that came from myself. I have this disorder, beta thalassemia. It's pretty common in the Mediterranean area. So my red cells are smaller than average, but I have a perfectly normal life, because only 1 chromosome is affected by the mutation. But now, if my partner had the same disorder, then there will be some, non-negligible probability. Then our children will have both chromosomes with the mutation, and this results in I think 75% of premature death. So I need to test, and my partner and I need to test for this disease. So we show how to do it, again, in a privacy-preserving way, so that I don't have to reveal my entire genome, just to know whether or not both my partner and I have the mutation. And finally, we go toward more, let's say, recreational test, ancestry testing, responding to this increasing demand of tracing one's lineage and ethnical heritage. So we try to, there are some companies that do that, Ancestry.com and 23andMe. They try to tell you how close you are to certain individuals, trying to find your ancestors and so on. And there are certain tests that you can do. You can trace mother heritage using mitochondrial DNA, some specific region of the genome, or father's heritage through the Y chromosome. But you can do very similar things, like assessing some similarity between two genomes. So we just look at a couple of them. One was Jacker similarity, and this involves only requires the computation of the intersection cardinality and the union cardinality. So we can do that with privacy, and we can do that efficiently. There are some algorithm tricks that were actually quite interesting. So we have some data sets and some kind of tests that we run on. Again, I'm going to skip the details for the interest of time. So that was also a small performance evaluation. But now, I just want to conclude my talk by looking at next steps. What we have to look at in order to bring privacy-preserving genomic tests to reality once really the majority of people in the developed countries at least will have access to whole genome sequencing. So the first question that we try to assess whether or not it's feasible to consider mobile devices for this kind of test. So like smartphones or dedicated devices, because it's much more likely that patients or individuals will let's say, carry a smartphone or a dedicated small device to the doctor rather than their desktop, right? And for the reasons that I presented before, it might not really be a good idea to outsource genomes to, let's say, to the cloud, even if you encrypt it. So we want to, test the boundaries and try to see whether or not this would make sense. And in order to do that, we actually go and implement a complete open source framework where we support pretty much all the pre-processing that is needed in all kind of tests. So usually sequencing centers give you sequence genomes in it's called raw format that you have to process in order to make it accessible for running these simple algorithm tests. So we do that, and we have a paper at WPAS last year where we release a toolkit. As I mentioned before, we also evaluate usability of applications built on that, and we provide a sort of API. So let me give you an overview. An API for application developers to build on top of this framework, so that they can implement their own privacy-preserving tests. So as more tests will become available in the future, we need more development. So we actually divide the framework into two parts. I don't know how to go back, but anyway. So the one part is the pre-processing that can be done at the sequencing center on a desktop, and the second part is what runs on the mobile device. So you can have a compressed, encrypted version of the genome, and then access it in such a way that you can run privacy-preserving tests. So we also have a small user study. We try to assess whether or not users will like to deal with these applications. So we even have an application on the Android market that simulates a paternity test. Of course, there is no real genomic information. It's just a toy application you can play with. And so the usability was pretty high. It scored 82 out of 100 on the system usability scale. Usually 80 is considered, everything above 80 is considered very good. But also it was interesting to see that interviews showed that users were actually concerned with privacy. So they in favor of genomic tests only if they do not invade their privacy. So just to conclude and leave space for some questions. So in this talk we have presented some analysis of whole genome sequencing. We have explained why this is such an important revolution in healthcare. So the good news. The bad news is that there are certain serious privacy and security concerns. We try to start answering some of these challenges by looking at the feasibility of using cryptography to support privacy-preserving tests on whole genomes. And the results are pretty encouraging and shows that using efficient protocols, but involving experts in bioinformatics, you can use domain knowledge and have good results. But our work does not end here by any means. There is a number of open research issues, and so we hope that more people can try to tap on that. And before getting your questions, let me thank my co-authors, a number of people that have worked with me. And thank you very much. Okay, so check.
There's a question. I have a question.
Yes.
Okay, so you mentioned that a couple of papers have shown that anonymization of genomic data doesn't work. Mm-hmm. But which type of anonymization techniques were they using?
Simple removing identifiers and removing some of the so-called SNPs. That they were believed to reveal ethnic heritage.
But do you think that, depending, of course, on the type of, for example, machine learning you have to do, that you may use a differential privacy techniques?
Yeah, I mean, it's definitely
better?
one interesting direction to explore.
Yeah.
I don't have a strong opinion on this. I just think that it's very hard to know in advance because we actually don't know much about genomes yet. There's studies that show new things every couple of months. So maybe we think that some parts of the genome are not, let's say, privacy sensitive, but in reality they will be in the future, right? So differential privacy can definitely help. It will be interesting to see what's the trade-off between utility and privacy.
Yeah, in particular, because there is a class, when you want this data to be used for research, mm-hmm. That's when they have to do association, as you mentioned.
Right.
In that case, secure set intersection techniques perhaps don't help.
No, don't help at all. Yes,
yeah.
Exactly. And so I actually talked to a couple of people in NIH, and for them it's a really open problem because they would like to fund more research on these genome-wide association studies, but they don't know how to protect privacy and protect them and volunteers from lawsuits. So it's a really hard question. Challenge. Yes.
So my question actually is not about privacy preserving situations, but are you aware in that literature without privacy preservation of the need for justification for an algorithm for questions of the following type, that you're given a bunch of sequences, mm-hmm. and you ask for like the two closest or the two farthest from mm-hmm. each other, things that appear to require quadratic time mm-hmm. In the number of sequences, and what's known about that?
So you mean the two closest genomes?
Yeah. So I give you a bunch of them,
uh-huh.
and I ask you, give me the two that are farthest apart,
uh-huh.
or the two that are closest
right.
to each other.
Yeah, actually.
Have people done that?
No, I never came across that. Actually, that's a
Well, do
very
you think that it's
interesting.
justifiable
Yeah.
from a practical point of view, so that we, somebody doesn't get rejected because of a very nice algorithm, but no one cares about that?
No, I actually think, I mean, on top of my head, I think it will help these companies that provide you with information about, your heritage, and they try to find your ancestors and things like that. So I think it might actually help them a lot, because so they have databases of genomes, and they try to tell you, oh, I have this person. You might not know that you're related, but you actually are. And so if both users have agreed to disclose this information, they will actually tell you. And this is a booming market, actually.
It's easier though than what I'm asking for, because
Yeah.
it's easier because, you come to me and so it's a query. Really, it's a nearest neighbor query. And,
Right.
you're giving me yours, and I'm telling you, okay, this is the nearest to yours or the farthest to yours, or something like that. The one I'm asking for is really the nasty one, because this is any two that are, farthest from each other,
Right.
or
Farthest,
closest,
I
or
can.
closest to each
Yeah.
other.
No, closest. If you don't know who you're related to, you might need to know who are the two closest to you, right?
Yeah.
So, in the database.
Are the genomes of identical twins exactly the same?
No, not exactly the same. Yeah. I don't know.
Do you remember
which
the
degree.
difference between them?
No, I don't think there is an exact number. There is a lot of non-deterministic ways that chromosomes are passed on between twins. But yeah, they're pretty close, but not necessarily exactly the same. I think it's so. As far as I remember, I think there is they're closer than, let's say, brothers and sisters genomes are, but not identical. Yeah.
Thank you.
Yes.
So my question is basically upon the fact that have you had a situation where you had to reveal more information, or rather expose more information in the sequence when you had to increase the accuracy?
I mean, I don't run this test, but
I'll give you an example.
yeah.
Let's say you, for your paternity test, you had an alleged father.
Right.
You're trying to map it to the kid,
Mhm.
and he has a sibling, a brother or something, and you want to be very sure that
Mhm.
the father is this.
Right.
I do not have much idea what
Right.
sequencing, so.
No, that's a very good question. So that's actually one of the reasons why at the end we really went for the simulation of what is done today in lab. So this test has been, many people have worked on, and they made it in such a way that it's admissible in court. So it's accurate enough that it will also capture the fact, let's say, alleged father and alleged father's brother, genome. They may be similar enough, but still not enough to cause a false positive. Yeah, so that's one of the reasons. Yeah, and so that's why we. Yes.
I have a couple of questions. When you spoke about releasing certain information to doctors, how would you go about doing that? If is the entire genome going to be cryptographically secure, and are they breaking it up into parts, or I don't understand that.
No, actually, the entire genome will be cryptographically protected, and that's actually, that's the only way where you could really protect privacy, provably, right? Yeah.
So then how would they give just a part of the genome to doctors?
Right. So the way it works, just to give a more concrete example, let's say you want to test for a certain kind of mutation. So the cryptographic protocol works in such a way that everything is encrypted and can only be decrypted if the test is positive. Right. So it's let's say a special kind of encryption where data can only be decrypted in case there is a match. So the doctor will not see anything, but only what satisfies his test. Right. That's. Yeah.
I just have one more question. It was
Yeah.
about the compatibility testing on slide 28. So you spoke about, oh, can you go back
Maybe,
to that
yeah.
slide?
I don't know why. Yeah. Yes.
There it is, yeah. This beta thalassemia, so if two individuals have it, each one has one chromosome, and they can still, they only have a quarter of a chance of having a child
Right.
that will die, right?
Exactly, yes.
50% chance having a child that will have the same one chromosome, right? And then
Approximately,
there's
yeah.
still a 25% chance that they'll have a child that has no, I mean, that both chromosomes
It's
do not have
approximate,
the.
yeah, but that's
So,
yeah.
I mean, like, doing this compatibility testing doesn't really protect anyone. And then also, they still have the same exact odds of having a child without it than they do having a child that.
So I'm not sure I got a question, but let me clarify what I meant here. So first of all, the probabilities are, let's say, rounded because certain chromosomes are dominant, so they are inherited with more with a greater probability. But yeah, so the probability that you will have a child with the major variant, so it's 25% approximately, right? So still it's not negligible, right? So it's something that you have to do. So I'm not considering privacy yet, right? So it's a test that it's better to do. Also because there are, I mean, I'm not sure for beta thalassemia, but for other kind of diseases, there are actually ways to treat, in uterus. Yeah. So, but in terms of privacy, so that I mean, you can be okay to just reveal to each other that you have this disease. So the privacy might not necessarily be a concern, but I mean, there are certain diseases that you might be okay to reveal to a potential partner, some that you might not, or if really these kind of genetic informations are used maybe in the future in online dating services, then maybe really you don't want to disclose it to unknown people, right? So there are different levels to that, but I mean what we really wanted to show is that if you want to do that with privacy, then you can. Yeah. Yeah. Thanks.
Sorry, I thought that what she asked, and she can clarify, is if the privacy-preserving comparison has exactly the same accuracy as the one. Is that kind of what you asked?
Oh, sorry, I didn't.
Yeah, I think that was.
I didn't get that. Yeah, it's exactly the same. So here, these tests are really looking at certain mutations. So in a specific position of a specific gene. So what you do is really try and look whether in this position, in this gene, you both have an A instead of a C. So the privacy doesn't take anything out of the accuracy. Yeah. Yes.
The goal is to have certain set operations be, or as the owner of your own gene, you can allow certain set operations to be done on your gene, so certain tests can be learned. But you also want certain tests not to be learned.
Right.
Have you looked at combinations of tests that could be learned that could infer?
Yeah,
the ones
that's a
don't
very
want?
good question. So that we, let's say, leave as an open question. So what we take the approach of having FDA approve what kind of tests you can do. So even if you do a combination of tests that potentially are disclose more than you expect, there will be something related to your health, right? So it's something that that you are disclosing. So the test will only reveal if there is a positive, right? So you run some tests and you disclose whether or not you have, let's say, a certain kind of disease or something like that. So even if you learn more than one disease, is something that you're willingly revealing, right?
If you're doing, say, compatibility on a dating site,
Yeah.
it seems like somebody could make up their own chromosome and make it constructed in a way that they're learning a lot about you.
Uh-huh. Right. So that's
even though they're just supposed to learn.
right. So that's so in the personalized medicine approach is we introduce this certification mechanism, right? In the compatibility test, we don't look at it yet, but so we envision a way where the sequencing center is actually signing these bits that are the sequence genome. So in that, so that you can embed some sort of authenticity into the protocol, so that you cannot just modify your own genome, right? But it's a complicated issue, because now you have to assume that the sequencing center verifies your identity or things like that. Do we want that? I mean, and it's hard, right? And also, right now it's in order to sequence one's genome, you need a sample. I apologize, but you need a sample of saliva, which is pretty significant. So you cannot just take your glass and take. I mean, this will be legal, but you cannot take your glass and have your genome sequenced. But in the future, maybe this might not be the case anymore, right? So I'm sure there will be a market where the sequencing machines will cost like $50 or $100 and will be sold on eBay, and then maybe this will happen. So again, I could just swap my genome with yours, or so it's very complicated. Yeah. Yes.
I just wonder, when you use enzyme to break genome into different pieces, that means the fragment, I wonder that this fragment also has a sequence number.
No. So in the paternity test, right?
So when you try to compare the two genomes just based on
Right.
this fragment, each fragment independently.
Yeah. So yeah, we do that in the paternity test, and no, there is no sequence number. It's actually the test, the RFLP based technique is done in such a way that two fragments will never have the same length. So yeah, so there is no need to associate a number to it.
So the last question is, I wonder, does genome has a character like the period?
Like what?
Period
Oh.
means it's just after long distance, it's just a repeat.
Yes. That happens a lot, actually. Yeah, there are certain. So genome is actually we distinguish genes and so-called non-coding regions, and in the non-coding regions there is a lot of repetitions. So certain patterns that repeat over and over and over. Actually, myself and I think also biology experts, they don't fully understand what non-coding regions are used for. Some say that they're used to instruct how to code for proteins, but only a part of them. But so some parts they seem like repetitions, but yeah, we don't know why. All right, thank you so much. Thank you.